Hi students, and welcome to Year 11 Biology and Module 2, Organisation of Living Things. This is video number 20, and we're going to be investigating transport systems in animals and plants by comparing structures and components using physical and digital models, including but not limited to plant vascular systems. So in this video, we are going to try and focus in a little bit more on plant vascular tissue. Plant vascular tissue is basically an arrangement of multiple cell types in vascular plants that are referred to as vascular bundles. We talk about tracheophytes, which includes the pteridophytes, the ferns, the gymnosperms, which are the conifers, and the angiosperms, which are our flowering plants. And all of these groups are characterized by the presence of vascular tissue. It's only very, very small and simple plants such as the algae, the mosses, and the liverworts, the bryophytes, that don't have vascular tissue. And you can tell that because they're very small plants. They don't grow very large. They don't have that additional support that the vascular system also provides, as well as efficient transport. So what we want to try and do is understand how plants facilitate the transport of water, minerals, and the products of photosynthesis throughout the plant. So let's look at plant vascular bundles in just a little bit more detail. There's three main sections to a vascular bundle. The distribution of vascular bundles is different between monocots and dicots, which are the two subgroups of angiosperms or flowering plants, and also differs between the arrangement of vascular tissue in the stems of each of these types and also the roots. So I've got here a comparison of a dicot stem with a monocot stem, and important that you also have a look at the difference in the distribution of vascular tissue in the dicot root against the monocot root. And what you'll notice is some different types of generalized cells such as the sclerenchyma and the parenchyma, but also within these vascular bundles, the important transport tissues as well as the undifferentiated growth tissues. So we see phloem, which is always sitting pretty much towards the outside of the stem or the root, the xylem, which runs to the inside of the stem or the root, and then the cambium, which basically sits in the middle, which is that region that sits in between the xylem and the phloem. That is a form of lateral meristem. Now, meristems are regions in the plant where there is active mitosis, so there's active growth. Cells are increasing in number as the plant is growing. So the tips of the roots or the tips of the stems, for example, are apical meristems, and we see a lot of mitosis happening there as the plant continues to grow. These ends of the plant are the apical meristems. In the center here, where we see the plants not growing up, if you like, but growing out, if that makes sense, these are lateral meristems. So cambium are an example of undifferentiated growth regions that are going to give rise to secondary xylem and phloem. And often they will be responsible in roots and in stems for this strengthening of the stem and the root. That is the deposition of things like lignin that are going to reinforce the plant, are going to give it an additional strength. And it's one of the reasons that we can identify how tall plants can grow just with these reinforced transport systems. So I've talked about the lateral meristem or the growth region that's formed by the differentiation of parenchyma cells between the primary xylem and the primary phloem. And those two, the primary xylem and primary phloem, are produced by apical meristems when the plant is first undergoing growth and differentiation. So these ones are going to divide and produce new phloem towards the outside of the plant. When we talk about ring barking, that is one of the problems, one of the reasons why plants can die, because you're removing those outer phloem layers, which means that the sugars that are being produced by the leaves, or at least in the leaves, is not going to reach the roots. And it's also going to produce new xylem or secondary xylem and phloem, which is what we've talked about. Some nice diagrams you'll find distributed around the internet and particularly in the BioLibra online resource, which has got some really good pictures in there and also some excellent information. And so you can see how the plant in its early development is going to use this property of differentiation in the cambium cells to produce more xylem and more phloem and continue to grow as a result of that. So we can see primary xylem remaining in the center around the pith. That's usually going to be reinforced. That's going to be woody tissue. You can see the phloem is now moved right out towards the bark, if you like, the outside, almost the outside of the plant, and that's where those sugars are going to be transported. It's also where, if plants get cut, you can sometimes see the sap emerging, and again, that's the sugars that are being released, or at least being transported in the phloem, that's actually able to escape when we cut the tree in that way. So this is the sort of stuff that we're looking for when we're trying to identify the characteristics of vascular bundles in plants. And as I say, this is just a way of giving you a little bit of an overview of how these tissues are formed and how they might change over time. We previously looked at xylem and tried to understand the processes of cohesion, tension, adhesion, the difference between the sorts of forces that are going to need to be overcome as the plant is working against gravity to allow for the movement of water up the stems, up very, very high distances in a lot of cases, and the fact that it uses those processes of cohesion and adhesion, capillary motion, tension, osmotic gradients, all of which are going to help the water to be moving from the source, in this case, which is going to be the roots, up to where it is, well, mostly going to be released in the leaves, but where it's also going to be used for photosynthesis. Phloem, on the other hand, are a little bit probably more involved than xylem in some ways. They have a few of these key components. They have the companion cells that are very important, play a very important role in the movement of sugars. They also have these sieve plates, which link to different phloem cells together to form sieve tubes. So the key ones are these sieve tube elements, and they link together, joined by these sieve plates, to create these long tubes. Now, of course, the difference with this process, which we've called translocation, to distinguish it from the transpiration stream, which is what allows the water to move up through the plant. Translocation is the movement of the organic materials, primarily sucrose, but it will include some organic and inorganic materials. And in fact, the nature of the concentration of solutes in the phloem actually creates concentradients with the xylem vessels that may be sitting alongside them, especially in younger plants, and you get a lot of this migration of water through the osmotic gradients that are being created. So that can create extra pressure inside of the phloem. It can also mean that the material is moving both up the plant, so it may well be produced in leaves that is then going to be moved. The sugars are going to be moved towards, say, a flower or a fruit or a seed, but it's also going to be moving down towards the roots as well. So the phloem are a little bit more complex. They're active cells, they're live cells. Often the xylem, we describe the xylem as dead cells, almost like straws running through the center of the plant. But these live cells, these phloem cells, very, very important in transporting organic material throughout the plant. Finally, something for us to have a bit of a look at in class and have a bit of a talk about, a nice little diagram here that came from the Instant Lessons in Biology, Book 1 by Alan Burns, and that was in about 1997. Still relevant, important stuff now. But you can see a source and a sink. So a couple of questions to ponder. Firstly, in which direction are carbohydrates transported in the flowering plant? Where are the source of the carbohydrates in plants? Where is the sink of the carbohydrates in plants? Can you contrast xylem and phloem? And there's lots of ways that you may be able to do that. Highlight some of those differences. What's the purpose of the vascular cambium? And how does the distribution of vascular tissue differ throughout a plant? Thanks for watching.